Good day, everyone. On behalf of BioIT World's Global Web Symposia Series and our sponsor, ArcSpan, I'd like to welcome you to providing an end-to-end informatics solution for drug discovery and development. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. I'd like to introduce our presenters. First is Bill Russo, Vice President of Sales with ArcSpan, and our second speaker is Jeff Carter, co-founder and Chief Operating Officer for ArcSpan. Welcome, gentlemen. The presenter ball is yours.
Thank you, Elizabeth, and thank you to everyone who joined us today in our presentation on providing an end-to-end solution for informatics and drug discovery and development. Starting this afternoon, we'll give a brief overview of ArcSpan, and Jeff will be taking the lead on that.
Thanks, Bill. My name is Jeff Carter. Just give you a minute or two about ArcSpan. ArcSpan is really focused on providing customers in research informatics with a system that manages all aspects of their lab informatics process. A lot of what we will talk about today will be focused on managing external workflows, but a lot of our customers use our tools for internal workflows as well as external. And so we'll be trying to focus as much as we can on demonstrating that value that we bring to the marketplace. We were one of the first SaaS-based ELN platforms. We started in 2011. And as I mentioned, we focus on data management. We have been GLP compliant and have had customers operating in GLP for over 4 years. And our customer base is largely small biotech as well as pharma, and that sort of spans the gamut from very large all the way down into medium-sized pharma. Virtual is a place where we do exceedingly well, especially because there's a lot of external research management in that area. But really, as I said, we'll focus on that quite a bit today, and I think that, that is probably the broadest part of our deployments across our entire customer base, big and small. We are based here in Massachusetts. We're based in Southborough, which is just a little bit west of Framingham, and we have a very active presence in the Cambridge area, as well as a lot of focus on the West Coast, and we have an office in Japan that just, I think, passed its 5-year anniversary, so we've had a good footprint in Japan for quite some time as well. So the next slide will be just a little bit about myself. I'm a chemist by education. My trade is software engineering, so I've spent all of my time in this industry in scientific informatics, which I very much enjoy. I have a great time with it. I've been doing software for longer than I've been in the informatics space because that's been sort of the focus of my career. So I started in industrial software at Amazon.com and AT&T, and then moved into the informatics area, going into Cambridge Soft and taking ownership of the ChemDraw application on a global basis from the product management perspective. And then after 8 years at Cambridge Soft, I co-founded ArcSpan. So I have been managing customer-facing operations at ArcSpan for the lifetime of the company, and very glad to have Bill have joined us about 6 months ago. I'm sure Bill can fix the timeline when he takes over on the next slide. So thanks for your time today, and hopefully you'll appreciate our demonstration.
So simply speaking, we're defining end-to-end informatics as the process of the entire lifecycle process from requesting work from either an internal scientist or on another team or an external partner, managing that entire process of requesting, receiving data, approving data, and then closing out the request from that team, as well as capturing all of the information associated with that scientific process, approving that data in a way that allows for greater trust and transparency of the work that's being done, managing any of shipping of samples to and from, and registering and understanding where compounds or cell lines are stored to allow for efficiency in executing work. When we think about the issues with the current paradigm, the first issue that we run into is that work is being managed typically via email. So requests are started, updates are managed, and work is closed out through a series of emails and potentially in SharePoint. But largely the files that are going back and forth through these systems are Excel-based or SD files for the work. The issue largely becomes that there is no quality or feedback loop during the execution of a request, and scientists have no way of understanding what is being done at the time the teams are executing the work for them. That results in receiving the data and then going through a process of having to manually manipulate the data, perform quality control on the data, and then execute communications with the partner after the work has been done to actually get the data in a format that they can use. And our teams tell us when we start with them that process can be anywhere from 2, 3 to 4 weeks from data received to data available. It leads to high frustration of the scientists and high frustration of the partner network that companies deal with. So if you were to propose a new solution, the solution that you would need to be looking for would need to, first and foremost, simplify the process. Eliminate all of the steps that sit in it and outside in email or systems that don't allow for near real-time communication with teams. They would have to ensure that information from the request to the receipt of the request and the status update are managed in the same system, as well as provide the means for all data to be added to the platform and quality controlled at the time of data collection. That would allow the teams to get the feedback that they're looking for at the time of execution and streamline the entire workflow cycle. It also provide greater transparency in the planning with partners and the updating of work as new data comes in and changes need to be modified. So any solution would need to understand the fundamental differences of the types of information that scientists use on a daily basis. The data and requirements of data are fundamentally different between what's captured at an experiment level versus what's captured and managed either in a registration system or an assay system. But those differences should be abstracted from the scientists. Scientists should be able to come into a system, understand the work that's being requested of them, and the system should manage and streamline the process of getting that work request in and allowing the scientists to put the appropriate information into the system and gain that feedback as they're doing it. So today, what we'll do is we'll follow that process through our proposed solution. From requesting a compound through the execution of the work to registering the information and then performing quality testing on the compounds, Jeff will be walking us through the system to allow you to see how a system could work in this environment.
We get to the dashboard. Basically, this is just sort of the landing page for logins, and I won't belabor it. What I'd like to do is jump right into requesting a set of compounds to be synthesized from the CRO. So we'll just click Submit New Request under the Work Orders menu. And what you get here is a form that is completely configurable. So which fields are on the form and what their data types are, their status, whether they're required or optional, valid values, things like that are all configurable. We're just showing you something that we think is particularly relevant. So we can make a couple of data entries here just into the required fields. And you'll see that I'm being asked to make an attachment. Like generally, this is a literature reference or an overview of the synthetic pathway or whatever. So we can just drag and drop that in. And then down below, we've got a table where the compounds will go in that constitute this request. So you can enter data into this table manually. You can draw with Marvin.js or ChemDraw. I prefer using an SD file because you can just drag the SD file in, and if you have fields in the SD file that correspond with the column headers in the table, then the whole table will automatically populate for you from the data in the SD file. You can also see here that I've pulled in a reg ID field. So what's happened in the background is that these compounds have been searched in the registration system to see if they've previously been registered or not. Generally, the chemist will obviously have a pretty good idea of whether they're expecting compounds to be registered or not. If you want to check one out, you just click on it and it will pop open in the registration system and you can have a look. So that is, there are many different ways of doing that, but this I think is the most straightforward, where you can just get your hands on the registration number for the compound if there is one. You can manage this table in a number of ways, including reprioritizing rows. This is intended to be a top-down execution list for the chemist who's doing the synthesis, or multiple chemists. You can get the data in and out of the table quite easily. I've shown getting the data in. You can actually download an SD file or a CSV file if you like as well. We'll go ahead and submit the request. Obviously, all the form validation is happening, so required fields, allowed values, things like that, and then the request will be submitted into the queue, and I'll be brought back to my dashboard. So we'll go over to my dashboard. This is a table of all of the requests that I've submitted into the system that have not been processed. So for example, if I want to select by a specific project, I can just filter and get down to that project pretty easily. Filters are easily removed as well, so we can open the table back up. I'll just sort by date created so I can get the one that we just submitted right at the top. We'll open this up. So there's a couple of things that you can do once a request has been created, and I think one of the more relevant ones is actually repeating the request. So generally speaking, this will allow you to, repeat a request so that you can get most of those compounds resynthesized. You can take some out, change the purity, do whatever, but it sort of simplifies the data entry. It also creates a link back to the request from which you made the copy so that you can see the chain of events a little bit more easily. So one of the things that we had this configuration set up to do is to create a new notebook in the system every single time a request is created. That's not something that has to happen, but generally it's nice to have all of the ELN pages that represent the compounds that were synthesized for a specific request inside of a given notebook. So I can click to open the notebook, and this is what either the internal or external chemist actually sees. They go to the notebook page, there's nothing in there. They can open up the request and take a look at the compounds that they're supposed to be synthesizing in a top-down order. So it's quite easy to copy these compounds out and synthesize one. I'll upload a known reaction so that I save you the pain of watching me draw a reaction in ChemDraw. And I'll do that by creating a new chemistry experiment. So this will create a new chemistry experiment in this notebook. You can see the chemistry experiment template. I'll upload a reaction. As I mentioned earlier, you can draw using Marvin.js or using ChemDraw. And of course, you can also upload reaction file, RXN file, SKC file, obviously CDX or CDXML, anything like that will work fine. So a stoichiometry grid is generated, a schematic of the reaction is generated, and we can start entering our data. So we'll enter some amounts for reagents. We can enter an amount that we actually got synthesized. And then, let's see, now we can go down. You can see my theoretical mass was 1.67. I synthesized 1.65, so calculations are done for you. In terms of write-ups, you can use standard blurbs to build your write-ups. So these are configurable by people who have permission inside of the company. You can see that the mass in the moles was brought in. If I go back to my overview and I make an edit, for example, you'll see that the data was kept synchronized with preparation. In terms of supplemental data, you can drag and drop files into the notebook very easily. So you'll see I did a selection and I'll drag these in. You can drag folders in. You can also drag nested folders in. The folder structure is preserved and it's so visually you can navigate the folder structure the same way that you dragged it in. So for these attachments, if you have an Office file or an image or a PDF file, the images can be inlined. You can view them inline. You can see that these are viewable as PDFs, so the PDFs are automatically generated on the server so that they can be easily displayed in your browser, regardless of the device that you're using. You'll notice that I haven't clicked Save yet. All the changes that I've been making are saved to a draft version, so that's persisted into the database, and you can, you know, the browser crashes, power goes down, whatever, you can always get back to your changes. You navigate to the page and it still shows up the same way. So I'll save this. We'll commit this revision of the experiment, and then we'll take a look at how to edit files. So if I go back to this Word file, for example, I can check it out. What checking out does is it opens the file in the editor that is designated on your computer for that file type. So obviously for a .doc file, it's going to be Word, but it might be Snapgene, or it might be ChemDraw, or it might be any other number of applications. As long as you've got that application associated with that file type on your machine, the live edit works very smoothly. So I'll check in my changes, and you'll see here that PDF will come back representing the updated document, and we'll just save the changes. So the live edit feature that supports the round-trip editing is available, just to reiterate, for any file type that you have an editor for on your PC or Mac. So we'll go back to the reaction now, and we'll talk about registration. One of the things that Bill brought up, which I think actually adds quite a bit of value, is instead of having SD files come back from synthesis and then registering them, you can allow people to register from the ELN directly. So we'll do that. So we'll go ahead and register this compound, and it's going to say, oh, you already have this. It's already registered as APR03. Put your data in, so we can do that. And you'll see here that we've also prompted the user for files. So if we want to get an NMR and an LC for each compound, we can do that. And I will upload those. These can also be attached to the experiment, either in addition to or instead of. So we're just trying to show some different options for collecting data. This way is nice because then you've got the NMR and the LC right on the batch record in the registration system. So it gives us batch number 54. I'll just save that off for future reference because we'll upload some data against this momentarily. So we'll save this to the experiment, and then you can see that the batch number rather is now linked into the ELN. So you can click through and get over to the ELN or to the registration system rather very easily. You can see the data are in that we entered, the amount, units, and purity. You can also see that you can view the NMR and LC. If you have, for example, an FID instead of an image, then you'd be prompted to download the file, and you can look at it in a viewer that's local to your machine. We've used images here to highlight the fact that if you do publish an image or a PDF or something like that, you can just view it in line, get to the data much more readily. And you can also see that it links back to the notebook page from which the batch was registered. So now that we are in registration, we'll move away from sort of the ELN workflow and into another common registration workflow, which is registration of an SD file. So if we go to bulk registration, first thing we have to tell the system is what kind of thing we're going to be registering. And I'll just point out that everything that I've shown you, we do equally well for biologics as well as small molecules. So we've taken a focus on a small molecule workflow here in this demo, but about half of our customers don't actually do any chemistry at all. So we feel like our value proposition is just as strong in biology as it is in chemistry. And you can see that, you know, we allow registration of cell lines and pretty much any other biologic that's common as well. So I'll register this SD file. And of course, it's going to ask for mapping between the fields in the system and the fields in the file. You can store these. So generally speaking, you'll have one per partner or one per vendor, or however you want to do it, one per supplier. And you can pull that down and then just let it go. So what's happening here is making a pass through the file to tell us if any of these compounds are already registered. So generally speaking, when you put an SD file into the system, you're kind of expecting that you're either adding batches or that you're adding new compounds. So usually it's sort of a surprise. It's a little bit of one and a little bit of another. So you'll see here we've got 20 duplicates. Everything in here is already registered, which is no surprise because when we registered or when we requested the compounds, they all had a registration ID. So this is sort of a QC check just to make sure that everything is good and we know that we're just adding batches and there's no new compounds in there by surprise. So we'll go through, we'll add all the batches to the compound records. If you've got a large SD file, you'll be notified by email when the processing is complete. You don't have to have your browser open. You can shut your computer off and go home and hang out with your kids. So once this is finished, we'll let that go through. Great. And let's just do a quick search, make sure those went in. So I'll do a search and we'll just say date registered is today. So we've got 20 compounds. And I want to show sort of the round tripping of the SD file because it doesn't happen very often, but it does happen that you put in an SD file that you didn't mean to. So if you have permissions, you can roll them back. So what we're looking at here is a list of all the SD files or bulk files that have been loaded into the system, Excel in the case of biologics. And we can roll these back. So it will say, here are the records that you're trying to roll back. Yes, I want to. Are you really, really sure? I'm really, really sure. And then it will go ahead and roll those back. If we rerun our search, you'll see that we now have 0. So we've shown putting compounds in by bulk registration, SD file, and then making sure they actually went in the way that we wanted them to, and taking them back out in case of emergency. I think we'll move on to inventory next, but I wanted to just take a pause and see if Bill wanted to revisit or re-emphasize any of the topics that we've covered so far.
Jeff, I think what we've done so far is we've executed the initiation of a workflow and then had data go into the system as an experiment and be captured by the system. If we were looking at how would the sponsor site see where the data was, how would a sponsor understand the data going into the system at the time of collection? Is there a project view or a notebook view that they can see the data coming in?
Sure. So there are a number of different ways that that can happen. The workflow tool, which is the very first tool that we interacted with, has a very rich set of notifications that can be configured with them, and that includes notifications when specific fields change. So you can see when a specific request is started or when it's completed or when data has been added, whether or not it's fully complete, by configuring those notifications. And those notifications can come by email, and they can also come in the system with the little browser chiclet. So that's the first way. The second way is that you can, of course, run searches on, most recent records changed every morning and look at the work that has come in overnight. So that's also an easy way to do it. And then that's kind of what I showed with the registration system was, hey, show me records that were created today. And then the third way, which just in the interest of time, we won't go into a lot, there's a feature in the notebook that we built for one of our larger customers that's actually a messaging system inside the notebook so that the collaborators can ask questions of the people who are running the programs, running the projects, and they can also get responses that way. The effort there, or the reason for that, is to move all that messaging from email into the system so that it's searchable, and that it's also, the message string is associated specifically to the experiment. And we use @ symbols to name people so that you can mention people, and anybody you mention in that comment is going to get an email that there's a new comment in there with a clickable link so that they can go to it. So obviously those messages are available to people who can view the experiment, but it gives you a way of targeting your message at specific people so that there's not too many emails going out, but you can get your question to the person that you want to. And obviously all those things are searchable in the system. Does that make sense, Bill? Does that answer the question you were asking?
Yes, it does.
Okay, so we'll talk very briefly about inventory. So now that we've registered some stuff, we probably want to be able to keep track of it and know where it is. So what I'll do is I'll actually enter some just commercial inventory. Our inventory is currently in use for commercial reagents, proprietary compounds, and development activities all the way up to and including GLP. So I'll show you, just in the same way that we talked about registration, how to get data in and how to get data out. If I go to my hood here, you'll see that it's empty. I can import. Again, we'll use an SD file. We'll make a bunch of bottles. We have to do a mapping just like we did in registration. And then off we go. The bottles are being created in the system. And then once that's done, we'll refresh the tree. So you can see we've got our compounds, we've got the detail information, and we've also got an audit trail. So standard inventory functionality, sampling, which is the same as aliquoting, with full chain of custody and lineage, editing of the record, using moving, check-in, check-out, and also a copy function. So what we'll do in order to round trip is I will show you how to get data out of the system or do other things, other operations using barcodes. So I have a set of barcodes here that we can use. So if you hover over the bulk operations menu, you'll see the different operations that we allow in bulk. And by bulk, generally speaking, we're talking about barcode scanning. But you can also do this by copy pasting values from an Excel file or just entering them by hand. So we'll bulk dispose all of the containers that we just put in except one, so that you can see that we're not sandbagging the demo. And we'll navigate right back to there. You can see there's one container left remaining, and you can also dispose that manually. So just a quick look at inventory. It's quite easy to integrate the notebook and or the registration system and or the assay system, which we'll move into next, with the inventory system. But for the purposes of this demo, we're going by batch numbers instead of by specific container numbers. Go ahead, Bill.
So Jeff, no, I just wanted to highlight the locks on the left-hand side and how they represent locations that have security on them. So only groups that are allowed to view or manage inventory in specific locations, they have to be added to those locations so that if you're working with external teams, they can manage their locations and update other locations, but they can't see everything that is within your inventory.
Correct. And just to clarify, the permission controls on these objects is per user and or per group. So you can give a group permission, but also put one user, add user by user as well. Okay, so we'll move into Assay. So we're going to run some tests on some material and then report the results back in against the batch number that we generated, that we registered, and then we'll be able to look at the results in the search tool. So again, just in the interest of making sure that everyone on the call knows what they're looking at, we'll go over to the search tool and we'll take a look at a canned report called a batch data summary. So we'll be pulling back these data for a given batch. You can see I have batch 2 pre-populated, so we'll run this search and you can see that I've got qPCR data and EC50 data for batch 2 that has come back. If you want to see information for full compound, all batches, instead of just one batch, you can do that very easily, and that will come back as well. So what we'll do now is we'll take a look at batch number, I think it's 54, that we just registered, and run this search, and we should get nothing because we haven't put any data in yet. So let's put some data in. We'll show you 2 assays today. We'll show you a qPCR assay and an EC50 assay. We'll run them slightly differently. So for each one of these assays that I've got set up in the demo, and we'll run 2 of them, I've got 2 protocols, an automated protocol and a manual protocol. The manual protocol is if you worked up your data outside of the system and you're just uploading the endpoints for availability in the database. And the automated one is you're going to upload raw data. The data workup is actually implemented in the system. And then the final data are published along with the raw data into the database. So for the qPCR, we'll run the manual. We'll add a result set. And for this one, all we're getting is the raw file. So this is a really simple protocol. We'll get the raw data for archival purposes. So the way that I like to think about this is that even though you're uploading the endpoints, you kind of want the raw data in case you want to reprocess it. So having it all in one place in the same result set where you've got the data that you're using to publish, but you also have the raw data, it makes it really easy to go back if you find something's not working the way that you think it should, if you find that out. So we'll save that. The data are being parsed and published into the database. Now, we'll go back and do the EC50 as well, and then we'll run some searches. This one is finished. We'll come up with a QC view, and we'll take a quick look at that as well. So we've got the reg ID, and then for each sample, of which there are 5 that you can page through, you've got your well-positioned sample ID, CT and CT mean, as well as library name and average size. So let's go back up and start upload of EC50, and we'll run the automated protocol on this one. You'll see that the protocol is substantially different than the one for qPCR, not just because the assays are different, but because there's a lot more information that we need to collect from the user in order to actually work the data up, as opposed to just collecting the endpoints and publishing them into the database. So we'll give it a name, and then we'll put the data in. Okay, so we'll upload this file. Now you can see that I've got top concentration dilution factor. These have default values, but they can be overridden by the user. And then we've got different starting point assumptions for the curve fit. I've put in basically all of the fit parameters, not that you would generally let a user change some of these, but I just like to illustrate the fact that you do have full control over all the parameters of the fit. This assay needs a cell line list, so I will put that in from Excel. And then I need my reg ID again. Now, in both of these cases, I've got the registration ID being entered manually by the user. That's not very common. I think usually you'll get the batch ID out of the file itself. But for purposes of just making things easy to demonstrate, we always have it so that we put the reg ID in manually so that we can register something new and then enter data against it. So let me rerun this, and you'll see that our qPCR data is now in the system. So this is all reportable now. You can download these results as an Excel file if you like, and then be able to do whatever you would normally do in Excel. And so it's fast and easy, and you can get back to the reg number as well, which will bring you to in a new tab. Let's go back and check out this EC50. Okay, great. So here's our QC view again. And you can see that since this is a plate-based assay, we get a curve and a data summary for each compound, but we also, for each concentration, I guess I should say, but we also can heat map these data. So you can, it's a fully interactive heat map. You can knock points out and back in from the map, and similarly from the curve. And you can change your starting assumptions on your fit and refit as well. Once the QC is done, you can save your results. And then we can go back in and rerun the search. And you'll see that the EC50 data now, along with the qPCR data, has come back as being reported against this compound. So one more time, Bill, I just wanted to give you an opportunity to fill in the blanks if you feel that there are any.
So Jeff, if we go back to the search and close the table view a bit, I think it's important to note how these reports were built. On the left-hand side, all of the values that are being captured inside the assay system are available to consolidate and search on or create a report on. That information can be combined with information from the inventory system, the registration system, and the ELN. So this view that you're showing will provide a means of seeing everything that's been done on a entity, whether it's a biological entity or a small molecule, and surface that information so people within a project can see what has been done, what is pending, and look at the results and make decisions in real time.
That's correct. And when I build these searches and save them, I can choose, for example, this is already a saved search, so I can choose if I want to save it as a new search or save changes to an existing one. I can also specify it as private. So by default, the searches are public so that you're basically making something that's supposed to be for the greater good. But you can keep them private, and you can also auto run them when they load. So for example, this search that I ran, I generally want to change the batch number so I don't auto run it so that I can parameterize the search before I execute it. But if I have a search that I know I just want that search to run every single time I open the page, you click auto run, you open that up and select the search. It will execute as part of the loading process. So that is everything that I had intended to show today. So I think, Bill, I will pass it back to you and to manage the rest of the process. And thank you, everyone, for your time.
So, Elizabeth, I think at this point we can look at taking some questions.
All right, we have several. The first question, how is data from an external team managed? Will everyone see everything collected?
Jeff, do you want to take that, or should I?
Sure, I can take that. So the permissions in the system are quite robust. So if you want everyone to see everything, then they can. And in fact, because I don't know who the person who asked the question was, generally, if you're at a small biotech, that is what we see most of the time. But at larger companies, you're going to want to probably restrict visibility of the data by either role or by project or both. So as I mentioned at the beginning, the permissions in the system in terms of who can see which data are at several different levels. They can be applied at any or all of the project level, the notebook level, the record type, i.e. chemistry experiment, work request, et cetera, or also the field level. So for example, you saw in the project dropdown at the very beginning of the demo that I had a list of targets, and you might not want your partner to see those targets, but you want to have them in the database. You can give internal users, whether it's by user or by group or by project, visibility to that piece of information, but not give it to anyone else, so that when a partner navigates to that record, they don't see a blanked out field, they just don't see anything at all, whereas when you navigate to it, you see a project, you see the project. There's even a little bit more gray area than that, and I appreciate the question, which is you want to see your target name, but you want your partner to see ABC123, some kind of alias. You can build that aliasing right into the configuration so that that happens at display time. It decides who is the user, what permissions do they have, and what do I show them? So for some users, it might show nothing. For some users, it might show the actual value, the real project name. And for other users, it might show an alias value. And that's all done under the covers in the configuration of the permissions and at display time.
All right. The workflows that you showed were using a small molecule example. Will the system work with antibodies, cell lines, and so forth?
Why don't you take that, Bill?
Sure. There isn't a specific requirement on either large molecule or small molecule. So as long as we can define what a large molecule is, like an antibody drug conjugate or a protein or a DNA, we can understand what makes that unique and register that into the system. Provide the same end-to-end workflow on that, the experimental data capture, as well as capturing the performance measurements like Jeff showed for qPCR and EC50 data.
Is it possible to have an approval process before a request is submitted to a CRO?
Yes, it is. And in fact, with a couple of extra minutes, I could show you that because that process is configured in the demo system that I did show. Basically, you can have a group of internal requesters and have those requests bottleneck through a group of people who manage the sequencing of those requests and or approval of them out to the CRO. And that is a separate page called the Manage Requests page that you can actually use to prioritize at the request level and approve at the request level. When the manager at the CRO logs in, what they're looking at is a view of the requests that have been assigned to that group and approved for them to work on. So there's a couple of different ways that can work, and you might actually be a little bit surprised at the different permutations that we've seen, one of which is that you can submit and those do go directly to the CRO, but then someone else can come in and override them all the way to both of the bookends, which is the Wild West on one side and only things that are approved on the other side. And any of those are possible. Through configuration. I'm sure some of the people on the call will appreciate my distinction between configuration and customization. We haven't shown anything or talked about anything in this webinar that does not fall under the definition of configuration and which would require customization or actual development effort.
Stephen asks, would a company that already uses an existing ELN have to adopt ArcSpan's ELN, or can ArcSpan be integrated into existing systems?
Bill, you want to take that?
Sure.
I guess that's a no.
The answer,
okay, go ahead.
The answer to that is you don't have to adopt the ELN to be able to take advantage of the workflow execution system, registration system, or the capabilities of managing the performance data within the organization. The one workflow that Jeff showed early on was having an individual register a SD file as a bulk entity. We showcase that typically because we do wind up having customers that have a notebook that they use internally and either nothing for an external notebook or having their CROs use their own notebook. So being able to provide the workflow, being able to capture the information that goes against the work request is important to us, and we can manage that independently of the notebook.
I'll add to that each of the modules that I showed, namely workflow, ELN, registration, inventory, and assay, are available on an a la carte basis. So we do have a number of customers, for example, that have internal registration systems or inventory systems that we've integrated with either into the workflow tool or the ELN or both. So these can kind of be mix and matched. And since the system is relatively new in terms of its architecture, it's well modularized and usually fairly easy to accomplish integrations. We've done many, so we may have already integrated with what you're thinking of. But even in the case that we haven't, it's usually a fairly straightforward effort to do so, although it can and occasionally does require customization, just to go back to my previous definition.
How does the system manage files from Prism or SnapGene?
So I'll take that one. A couple of things about that. We can put under the umbrella of Prism or SnapGene basically any file type that isn't an Office file or a PDF. And so we have a lot of users, including a couple of our biggest customers, using SnapGene specifically, and obviously pretty much everyone in biology is using Prism. So those files can be attached to into the system by drag and drop, as I showed a couple of times during the demo. And then the live edit technology can be used to round trip edit. So that means that you basically check out the file, the PZF file, and it opens in Prism. And then you make your changes and click check in, and the changes are persisted. And then the audit trail is kept for who made the change, what change was made, i.e. what's the difference between the two files, and when was the change made. Same exact thing goes for SnapGene. And as I mentioned, we have a pretty significant user base using both of those tools in the method that I described. Bill, I want to take a pause and see if you have more to say about that, because I know that this is an area that you've been particularly active in recently.
No, I think you got it. I think what most people want to be able to do is go from a file stored in the system to being able to work directly inside the best of breed tool. So in the case of SnapGene, being able to evoke SnapGene directly from our platform, or in the case of Prism, being able to launch the Prism solution directly from our system and have all of the information version controlled, I think is what the individual was looking for.
Can your system integrate with an internal compound registration system?
Yes, and we've done that actually a number of times. The best way that you can get information about that from a non-biased source would be the white paper that's available on our website that was written by some of the people that we work with at Synovian Pharmaceuticals, which is a Massachusetts company, and we have had an integration with their behind the firewall Accord registration database for about 5 years now, and we enable registration of batches by their CROs directly into their Accord system. So that is well documented in the white paper. There's also a recent user group meeting talk that was done by those guys about exactly this topic. So we can provide all of that to you. You can also self-serve off of our website.
Thank you. Just out of curiosity, can you push files to another system like Viva?
We can. We do that currently into a number of different systems, including SharePoint, Documentum, and Ignite. There are a number of different ways that we can do that and a number of different business requirements. It's sort of a spectrum. So I think I'd want to know a little bit more about the specific use case to comment further, but those are, those are systems that we have active in production integrations with for movement of files between the two systems currently.
All right, thank you. Our next question, is the data uploaded from files searchable within the system?
Bill, you want to handle that?
Sure, I can take that. So the answer is yes. If we understand the file format, like an Office document, Excel spreadsheet, a PDF, when the data is loaded, that data is indexed and is free text searchable through this system. So that is a typical requirement of our customers, and we do it all the time.
All right, we have time for just one more question. In your presentation, you mentioned projects. What are projects within the system, and who can see projects?
Okay, that's a good question. So a project is basically a virtual kind of permission object in the system. It's a collection of references to either notebooks, experiments, registration objects, etc. So you can attach anything or include anything in the system inside of a project. The projects are visible to, and I'll air quote, people who have permission to see them, right? So for example, a system administrator will be able to see all of the projects. And then below that, you can have standard business rules or ad hoc per project configurations that allow different groups and users to be able to either see or edit the projects. So if you can see what's in the project, then I think that's fairly self-explanatory. If you can change what's in the project, then that means that you can add and remove content from the project. I think one of the more interesting permutations of permissions is if you can edit a project, but you don't have view. You can't see it, but you can edit it. And what that means is that you can look at data that you have attached to that project, but no one else, and you have the ability to manage those references. So that gives a user, for example, the ability to attach a notebook page to a project or remove a notebook page from a project without being able to see any of the other information in that project but their own. So those are sort of the three different project level permissions that are available.
All right, perfect. We're coming up on our last minute. So I'd like to take this opportunity to thank our presenters today, Jeff Carter and Bill Russo. I'd like to thank ArcSpan for sponsoring today's web seminar. Most of all, I'd like to thank those of you who came and spent this time with us. We're very grateful you came, and we hope you were able to find some answers to your research questions. So with that, on behalf of BioIT World's Global Web Symposia Series, I'd like to say thank you all so very much and have a great day. Bye-bye.